Hi, I'm Jeff Galvin, the CEO of American Gene Technologies. Thank you for tuning in today. Before I get started, I need to give you all the normal legal disclaimers. You can read that later, but basically it says that I'm going to be making forward-looking statements, and these are not guaranteed. So if you're not familiar already with American Gene Technologies, we are a leading technology company in gene and cell therapy. Now, what's unique about American Gene Technologies is that we have created a platform of intellectual property, of discoveries, of inventions, of components that are critical across many different disease areas of gene and cell therapy. You can think of them as almost like an iPhone for disease cures, where eventually what we hope is it'll be as easy as writing an app to cure a new disease, because the components will take care of delivering the therapeutic genes in the right levels, in a safe way, to the cells that need it. Because that's the heart of gene and cell therapy, is there are a lot of commonalities between all the different changes you might make to DNA in order to drive disease solutions. So a platform would be useless unless you could make drugs off of it. And of course, in pharma and biotech, you need to prove that it works before anybody is going to look at your components. And so that's exactly what we're doing. And we have lead programs in infectious disease. We have a cure for HIV, and that's going to be the main topic today. We also have a potential cure for phenylketonuria, which is a monogenic disease, of course, so it's the replacement of one gene in the liver to mitigate that disease. And then we have some exciting things down the road in immuno-oncology. We have a potential immuno-oncology asset that we think in 2023 could make it into the clinic, and it could reduce some of the most serious cancers out there: breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers, to one shot into the primary tumor, and the natural immune system will rise up to the level where it will clear all of the tumors. That would include the secondary tumors and metastases. So we might be able to send radiation and chemotherapy the way of bloodletting and leeches. That was the long-term dream of American Gene Technologies, but there's a lot of low-hanging fruit in the meantime, where the scope of our platform and the power of our platform should give us the ability to turn out cures to a variety of different diseases, because we have the right components, and it just needs a little bit of creativity and a little bit of construction or engineering on top of that to effect a cure. Now, the big news today is about HIV. So if you have been following us, you know this already. We are in the phase 1 clinical trial for an HIV cure. And the first 2 patients have already been infused, and we have seen 0 serious adverse events. And the DSMB, as of the shooting of this video, has seen all of the data for the first patient, of course, and it cleared us through with no changes to the protocol. And from what we're seeing in the second data set, which will go to the Data Safety and Monitoring Board before you get to see this video, it looks like it's similar data, and so we expect to get passed through again. And at the beginning of October, which will be again in the rearview mirror by the time you see this video, we will have infused the first patient. Hopefully by the time you watch this video, we will have already seen the 7th day examination of that patient, so we would have gotten any of the short-term adverse events. So that's where we are in this clinical trial. It's very exciting because you may know that phase 1s can give you not just safety data. This isn't like a typical pill or a shot, or something that goes all over your body like a monoclonal antibody or a vaccine or whatever. We don't have to give this in small quantities to healthy people to see if they get sick. No, all of the people that enroll in this study have HIV. It's well controlled on antiretroviral therapy. So they have a relatively normal immune system, and then we're able to modify that immune system to make them permanently immune to HIV. So we have HIV positive persons who have already been treated with the therapy that I'm going to describe to you now, and that have not shown adverse effects. And I'm going to go through a little bit of the reasons why I believe this thing has a very good shot on goal. I personally believe that this is going to work. And the beauty of a phase 1 trial on sick patients is you get efficacy signal, and you could even get first human efficacy. First human efficacy would be the step that would make it exceedingly likely that this could be commercialized. But don't let me get ahead of myself right now. Let's go ahead and talk about this so you can understand it a little bit better. So what do? I already told you that the patients that take this treatment will be well controlled on antiretroviral therapy. And what that means is that the body will have recovered from the initial HIV infection, and they will have the full diversity of their T cells, including HIV-specific T cells. So why, if you and I and all of us watching this video have HIV-specific T cells, unless you've got wasting disease where you've got HIV and your immune system has been devastated and you have full-blown AIDS, basically, if you're well controlled on antiretrovirals, or if you've never been exposed to HIV, you have healthy HIV fighting T cells in your bloodstream. The only problem is they're not quite good enough to actually fight HIV, because HIV virons have evolved the capability of infecting the sentinel T cell that is supposed to detect HIV and trigger the entire immune cascade against that pathogen. So it's very different from COVID or a cold or any other virus, even like polio, because even though you have T cells that are ready to fight it, those T cells are actually the first cells to get infected, because they run over to the pathogen, they try to detect it and start the immune cascade, but in the meantime, that viron will actually grip onto that T cell, merge with it, and implant the HIV genome in the sentinel T cell that was there to detect the pathogen and fight it. All right, that is the only reason that HIV gets into your body. And so the key to our process is we will find those HIV fighting T cells. They're specifically CD4 positive T cells that are HIV specific, and we modify them so that they can never be infected. Well, if they can't be infected, guess what? They can operate just like all the other T cells in your body against their pathogen. They can clear HIV like you clear a cold. This has been demonstrated already in many patients in the past, in the Berlin patient, in the London patient, in the Amsterdam patient, and even in a clinical study by a company called Syngamo that showed that they could do enough modification to get a small number of people functionally cured by modifying those T cells. Okay, so here's what we do. Our process is, I think, the next step on all of the research and all of the results that have been seen in this industry to bring this to a commercializable level. And what we do is we collect a leukopak from the HIV-infected individual who is well controlled. So what we get in there is a filtration of these T cells, and there'll be a very small quantity of HIV-specific CD4 positive T cells in there. And that's what we focus on. This is all done in an automated benchtop unit called a Miltenyi Prodigy. And what the Prodigy does is it takes the blood automatically through a series of steps where we go ahead and stimulate the mix that proliferates the HIV-specific T cells a little bit with a proprietary peptide mix. And then we go ahead and we do a depletion protocol where we reduce the non-HIV T cells to the minimum level possible so that we can save viral vector and make this process cheaper. And also we need room, because the step after that, after we modify them, we go ahead and take whatever T cells are left over, mostly HIV T cells we hope, and we modify them so that they're impermeable by the HIV viron. In other words, they are immune to HIV so they can fight it normally. And the final step is that we culture them up to 1 billion HIV-specific CD4 positive T cells that are uninfectable by HIV. Remember, the CD4 T cell is the conductor of the immune orchestra. It is what triggers the CD8 response and the antibody response to any pathogen. And HIV is absolutely the perfect virus for knocking out the CD4 cells that are trying to fight it. And guess what? It doesn't even infects CD8 cells. The CD8 cells go away because the CD4 cell stops telling them to react, and the antibodies go away for the same reason. So if we restore that protection, we believe what we can do is if you have enough of that in your body, you can be immune. The next step is we go ahead and there's a fairly long, for the clinical trial, about 60 to 70 days of release criteria. That'll be shorter in the final product. And once we determine that there are no contaminants and no replicating viruses and so forth in there, that it's safe, a safe product, last step is just reinfuse it into the patient. The amazing thing about this protocol is that none of it requires any inpatient treatment. In other words, the leukapheresis is outpatient. All of the blood processing is just done, on this unit that any hospital could buy, so it's transportable all around the world. And it's, one button and go. You hang the reagents on it. You need a specially trained operator, but it's not rocket science to end up making the blood product. And then once the blood product is determined to be releasable, any hospital can do the reinfusion. The reinfusion only requires a very mild cytotoxic conditioning protocol to eliminate some of the T cells in there. So the current protocol requires a treatment that will do a small reduction of T cells in the body, but in the first couple of patients that hasn't been painful. It's not like the kind of conditioning protocols that are being used in things like CAR Ts. Okay, so why do I think this will work? I think it'll work because in the preclinical data, we made a lot of these blood products. Now, without an IND, we couldn't reinfuse them, but we could test them ex vivo, and we could dump as much HIV into them and as many infected cells as we wanted to. And what we saw was a remarkable capability of that blood to clear itself of HIV and HIV-infected cells. It was so remarkable that we showed it to NIAID. They were also, may I say, amazed, but let's say impressed enough that they wanted to repeat that in their own lab. They actually asked us for a collaborative research agreement. We went ahead and signed one with them. We gave them just the reagents. They bled their own patients, and they got the exact same data. And they actually co-authored this article in Molecular Therapy. So the head of the HIV lab, Tae-Wak Chen, in NIAID, is on this article, along with some of his postdocs. And what it shows you is that it looks like the blood clearance capability is about 3 to 5 times what's necessary to functionally cure the patient. In other words, in a one-and-done therapy, we can convert that HIV-infected individual from having to take antiretroviral therapy to suppress the virus to be able to naturally suppress the virus at levels where they can never get AIDS, they can never transmit the virus, and they can never be reinfected, and they don't need any antiretroviral therapy to maintain that, we think, for life. So, when will we know whether this works? Whether it's more, it's more proof than just confidence and, sort of projecting forward on the data. We'll know that when in the clinical trial, we go through a treatment interruption protocol. And we expect to do that early next year. And so we should have some solid efficacy data by next summer. But we are also taking blood samples all along this process in parallel with the physical checkups and the normal blood tests for monitoring, so that we can find objective markers of efficacy that would be convincing to scientists. We may start seeing those around December. And we'll go back to NIAID and see what they think. So remember I was telling you that Syngamo did a study that, we had, we sort of look back at the Syngamo study as being another sort of proof of concept that the idea of gene therapy could convert people to functionally cured. Because in their study, they were able to get 1 in 10 patients durably in remission without antiretrovirals. That's a very important finding. Now the problem is they were using $500,000 worth of viral vector. They were only getting 1 in 10 patients, And to get into the study, you had to be what was called a heterozygous delta 32 mutant. And what does that mean? It means you're already missing half of your CCR5, which is sort of a latch point for HIV. So they had done a study basically where they had depleted enough of this latch point so that some of the patients got better. And what you see in our study is that we are getting, for 1/10 the amount of viral vector, the cost, 1/10 the cost, we are getting 2,000 times the dose of these critical CD4 positive T cells that will conduct, that will create a normal immune reaction in the CD8s and the antibodies, and also as CD4 cells, they act on the virus. We get 2,000 times the dose at 1/10 the cost, and it works in anyone because we're using a different technology to eliminate the CCR5. So we're eliminating that latch point, but we're also putting in 2 other short interrupting hairpin RNAs that are against conserved regions of all known genomes of HIV. So all the known strains, we find conserved regions, and we put knockouts for VIF and TAT. And what that does is it gives us 2 more layers of protection. So even if a virus doesn't need CCR5 to get into the cell, it doesn't matter, because if it gets into the cell, 2 of the critical genes for HIV will get knocked out by shRNAs that we've installed into the DNA of that cell. So we've got 3 times the protection of Syngamo, still 1/10 the price. Then the other thing that we have figured out in our cell process is how to get a very high percentage of the T cells actually modified. So we calculated that Syngamo got approximately 10% of the T cells that they tried to modify with their viral vector. They got 10% of them depleted enough of CCR5 that they were effectively immune to the R5 strain of the virus. It wouldn't have worked on X4 tropic strains, but on R5 strains, it would have worked. So 10%. Well, when we do this automated cell protocol, we're getting consistently above 90% of the cells, usually above 95% of the cells, transduced and modified and immune to HIV. So we're 9 times the reliability as well. So 2,000 times the dose, 3 times the protection, 9 times the reliability. They got 10%. What could we get? I think we might cross that threshold of functional cure. And now we have data that says that 2 patients have demonstrated that this is safe, and by the time you watch this video, we'll have a 3rd one that will have been monitored at least for 7 days for serious adverse events. If we get that, then we'll be able to pick up the pace of treating this and get a full data set together for the FDA, along with those objective blood markers that would give scientists confidence that this will work. All right, so what would I do if I was watching this video? Well, it would depend on who you are. If you're a partner, a potential partner like a pharma company, maybe this is a time for you to look a little bit more closely at this. This is something that's going to be a game changer in HIV. If HIV is a strategic area for you, we would love to talk to you. We're not averse to partnering this up. And this is really the time, when you think about it, that it's only, 6 months to, or less than 6 months to probably some blood markers, and less than a year to potentially first human efficacy, and that we have so much data in the rear view mirror that gives us confidence that we can get there. Okay, maybe this is the right timing. We've talked to a lot of you already, but maybe it's time for another look. If you're an investor, well, we can only talk to accredited investors. We are a private company, but if you're accredited, which is, you can just look that up on the internet. It's not, you don't have to have a certain license or anything like that. You don't need a certificate. You just have to be of a certain financial wherewithal that you can evaluate and purchase things like this. But if you're an investor, I'd go ahead and get in touch with us if you're accredited. And this is not a solicitation, by the way. and you need a lot more information to evaluate the company to see if it's right for you. If you're a patient, I think that your reaction to this video should be keeping your fingers crossed. I think there's enough data at this point that it's not unrealistic or unreasonable to feel some hope that we may be able to bring a functional cure to you and restore you to a normal life. And that is the mission of AGT. It's to use the power of gene and cell therapy to reduce suffering and reduce death from serious human diseases. And we know that there are over 1 million people, even just in the United States, suffering from HIV. And even if they're well controlled, even if they're undetectable with a standard lab test because they've been religiously taking their antiretrovirals every day, they're still suffering. They're suffering because it's just no fun to be different. There's a psychological burden of having that knowledge and being reminded of it every day with a daily pill. Don't underestimate that if you don't have HIV. That is a burden on all of these folks. But also, the antiretrovirals themselves have all sorts of side effects. They're way better than having AIDS. They're the right thing to do. But if we can get rid of the common side effects of antiretrovirals, because we can eliminate antiretroviral therapy with a one-and-done cell therapy that makes your immune system capable of fighting HIV, well, we can eliminate things like diarrhea, nausea, headaches, fatigue that are not uncommon with antiretroviral therapy on a daily basis. But there's also the long-term impact of taking these chemotherapeutics. Things like early aging, bone density issues, osteoporosis, brittle bones, liver, kidney, heart disease, and extra cancers. HIV patients are much more expensive to society than just a $20,000 antiretroviral therapy treatment or a $30,000 antiretroviral treatment. Insurance companies are putting out a lot of money treating the comorbidities of the low-grade infection and the side effects of antiretrovirals that makes these patients not only have to suffer and go back and forth to the doctors all the time, but it costs a lot to society. Every HIV patient, every newly infected patient, is probably going to cost society somewhere around $3 million, assuming that they seek out treatment and live a normal life. So this is a big healthcare burden with over 1 million infected people, 1.2 million in the United States, roughly 50,000 new infections every year. It goes up and down, but between 30 and 50, and this has been a heavy year. So this is an issue that we should all care about. We care about it deeply at AGT. This is our first therapeutic. We're feeling very confident that we can show that this thing works and that we can commercialize it and bring it to patients that are in need. And then the best thing about AGT is this is just the tip of the iceberg. We can reapply even the HIV technology to other chronic viral infections like hepatitis B, HTLV, herpes, to reduce some of the consequences of those chronic viral infections. But we also have assets all across the board that can help to power dozens and dozens already of monogenic disease solutions, like PKU that I showed you, and a couple of immuno-oncology assets that I think are real wild cards. And I'm very hopeful that we can send radiation and chemotherapy the way of bloodletting and leeches one day. I'm sure gene and cell therapy will do it. Gene and cell therapy with CAR-Ts is already doing it in some areas of liquid cancers. So, for anybody that cares about cancer, there's reason to be hopeful that the future's quite bright, and AGT has something major to contribute in that area as well. So we're going to focus on HIV, and then we're going to use the profits from HIV to go ahead and power some of these other solutions. But we've got an expanding portfolio and an expanding scope of our platform that could lead to a tremendous amount of good for patients in need. All right. Thank you for listening to me. And all of our contact information will appear right after this if you want to get in touch with us. I hope you'll at least connect with our social media and track our progress. Thank you.